Good evening. Welcome back to tuition2u.com. You are now with V. Sundram. And in this video, we are going to discuss about how to answer paper 3 question. The experiment that we are going to discuss today is about fermentation by yeast. I'm going to explain to you how you go about answering an unguided paper 3 question. Unguided paper 3 basically means how to conduct the experiment is not given. You are supposed to know how to conduct the experiment based on what you have in the textbook and also based on what you already learned in school. If you still don't have the question, you can download from my website, tuition2u.com. If you go to my website, is here, you can see the name, tuition2u.com. And you can just click on paper 3 course and you can register for free and download. I have 2 questions there. I have the unguided question, which I'm going to discuss now. There is another one, which is a guided question. The guided question is actually from trial paper for Terengganu. So you can download the question. That is the guided one. You can download the question, plus you can also get the marking scheme. I have both in my website. You can see. This video is to explain how to answer the unguided question. The unguided question answer I didn't give in a PDF file. You have to watch this video to understand how to answer it. So let's quickly go into the explanation. Okay, so if you already download the question, this is how the question should look like. Now I've recreated the whole question specifically for the trial paper. We don't know what's the question going to come up. If it's alcohol fermentation by the yeast, maybe the question will look like this. We don't know what is the question. Your best policy is to cover as much as possible. If it's alcohol fermentation by yeast, then how the question is going to be. In the textbook, the alcohol fermentation experiment is quite simple. When you conduct the experiment in the alcohol fermentation, ethanol is produced, energy is produced, and also carbon dioxide is released. So to detect the carbon dioxide, we can use lime water. If carbon dioxide gas is present, the lime water will become cloudy or become chalky. If you smell the mixture, you can smell alcohol. So if you can smell the alcohol, if the lime water turns chalky or turns cloudy, it proves that the yeast is doing alcohol fermentation. So the experiment that you have in the textbook is rather simple. We are just proving that the yeast is doing alcohol fermentation. What we are doing in this experiment is we are going to modify the experiment that you have in the textbook. What we are going to do now is we are going to change the concentration of substrate. We are going to change the concentration of glucose. By changing the concentration of glucose, what happens to the rate of alcohol fermentation in the yeast? So it's a modified experiment based on what you have in the textbook. So let's quickly move into the explanation. 2022 trial paper. Let's assume you are studying under my school, which is Sekolah Menengah Kebangsaan tuition2u.com. Okay, the paper should be 45 minutes. You can read through the instructions here. First thing you have to do during the experiment is the question paper will have a list of apparatus and material. Check through every one of them because they give you around 10 minutes or 15 minutes to check the apparatus and material. Go through one by one because we don't want to have any problems while we are conducting the experiment. Generally for bio, if there is any devices, make sure the devices are working. For example, thermometer or a stopwatch. If you have any mechanical device, make sure it's functioning properly. Other than that, just make sure all the other components are given. Test tube, for example, check whether there is crack or anything like that. So you use that 10-15 minutes to go through the apparatus and material. At the same time, while you are going through the material and apparatus, you can generally already know what is, how to conduct the experiment. In case, I'm discussing alcohol fermentation by yeast, but let's assume in the actual exam, some other experiments comes out. Even if you didn't study anything about the experiment, the material and apparatus is right in front of you. So based on the list of material and apparatus, you can more or less know what is the experiment about. So that's the beauty of doing the practical for paper 3. In the previous syllabus, they write the practical, they write written exam, and there is nothing in front of them. There's only question paper. And if they don't know, they don't know. That's it. They can't answer the question because there is no clue, nothing. For you, even if an experiment that you didn't know suddenly comes out, at least you have the list of material and apparatus. Using that and using some information from the question, you can more or less know how to conduct the experiment. So make use of this list and the list of apparatus and material to know how to conduct the experiment. So after I check through the whole list, what I'll do is I'll go through this and I'll highlight some of the information that I feel that is important. Normally they'll have some quantitative words. For example, here you can see there is 2 grams of dried yeast. In here, they mentioned there is 5% sucrose solution. They also mentioned there is 10% sucrose solution. So that tells me that the sucrose solution concentration should be the manipulated variable. We haven't looked at the question. From the list itself, we know the sucrose solution got 2 types. So that is the one you're going to change, 5% and 10% sucrose solution. 2-gram dried yeast. Every experiment, you're going to use the same amount of dried yeast because the value is given 2 grams. And we have some other information, paraffin oil, lime water. So when you see lime water, you know more or less you're supposed to detect the presence of CO2 gas, carbon dioxide gas. We have boiling tube, measuring cylinders, and glass rod. Okay, stopwatch. Make sure you check whether the stopwatch is functioning properly. This is the list. So just go through this. Once you're satisfied with everything is normal, wait for the instruction and you can start to do the question. So once they allow you to start the question, we go straight to the first question here. Okay, so the question. First sentence is normally general description of what you're supposed to do in the experiment. So yeast fermentation is a metabolic process in which glucose molecules are broken down by yeast anaerobically in condition of limited or no oxygen. Okay. Based on diagram 1, plan an experiment. This sentence is important because they are telling us what is the experiment and what are the manipulated and responding variables. So you need to take note of this sentence when you have the word plan an experiment, do or conduct an experiment. The sentence after that is important. So I'm going to underline this. Investigate, plan an experiment to investigate the rate of reaction of yeast. So that is the responding variable, rate of reaction of yeast. And in different concentration of glucose concentration should be the manipulated variable. So we can identify the manipulated variable and also the responding variable from the statement of the experiment. From there itself, we can already know what is the experiment all about. Take note of the diagram also because most of the time there is some information you can get from the diagram. Like in here, I can get 2 of the constant variable from the diagram. The first constant variable that we can get is all the experiment uses 15 ml glucose solution. So 15 ml of glucose solution is supposed to be constant. 2 grams of yeast is also supposed to be constant. So in the next experiment, this is test tube A, boiling tube A. In the next experiment, boiling tube B, what you're going to do, you're going to use the same amount of yeast. You're going to use the same volume of glucose solution. But what you're going to change is the concentration of the glucose solution. Okay, so basically almost all the information that we need, the basic information that we need for the experiment, you can get it from the statement of the experiment and also from the diagram. Once you already know more or less how what is the experiment all about, we can start to go to question A. So question A, plan your experiment by using the apparatus and material provided. Your procedure should include methods to handle the variable and also precautionary measures. They're giving you 4 marks. There is 3 variables, manipulated variable, responding variable, constant variable. There is also at least one precautionary measure. So that is the 4 marks. Whether it's a guided question or an unguided question, they are going to ask you identify the variable. How do you control the variable? The whole of paper 3. Now, one of the most important technique that you need to know is Make sure in every answer that you are providing, you have the variables, mainly the manipulated variable and responding variable. Whenever possible, we add in the constant. That's how you answer paper 3 question. Write the variable, identify the variable, and write the variable in every single answer. You can get your full marks. That is the trick. Variable, that is the keyword. Identify the variable, write the variable in every answer. You get your full marks. So let's look at this question. So based on the diagram and also based on the list that we have here, we understand that both experiments, we are going to change the sucrose solution percentage. First experiment, we use 5%. Second experiment, we use 10%. That is the manipulative variable. What is the responding variable? The responding variable is given in here, which is the rate of reaction of yeast. Now, how do you calculate the rate of reaction of yeast in the experiment? Because it's a calculation. To do the calculation, what do you measure in the experiment? So what we can do is, we can go to further question. Normally, the question like this is question A. If you go to question B or question C, you'll see a table. In the table, you can get the clue. Okay, if you go to this question. So there is a table here, which is question C1. Actually, by right, I should have made this question C1 as question A. So if you look at this table, you can see this word number of air bubbles released in 7 minutes. That's how you get the responding variable. Number of air bubbles released in 7 minutes, that is what we measure or we calculate or we count. In this case, we count. And normally, when you are mentioning the responding variable, you need to mention what is the device you are using. For example, if you say measure the time taken, so you must use the word by using stopwatch. If you measure the distance moved by the air bubble, for example, transpiration experiment, if you measure the distance, you must mention by using a ruler meter. The device must be mentioned, but this is a unique experiment. We are going to count the number of bubbles released. To count the number of bubbles released, basically we are going to use the eyes and look at the bubbles. So there is no device used. So unique experiment, the device is not mentioned. But if there is any other experiment that uses device, you have to mention the device when you're explaining about the responding variable. Where do I get the responding variable? I can get it from the table. Actually, even manipulated variable I can get from the table. So here we already have concentration of glucose solution, which is 5% and 10%. So look for the table. In the table, you can get the manipulated variable. You can get the responding variable. Use that to answer question A if it's unguided question. So if you go to question A, okay, the first thing we are going to do is we are going to explain to the examiner how we control the manipulated variable. We can just use this table. We can use back this table that we have here. Write back what you see here. That should be 1 mark for manipulated variable. So if I come back here, what I'm going to do is my first answer is this. So use different concentration of glucose. So we mentioned we are using different concentration of glucose. And take note, you must start the answer with the word use. Without that, they might not give you the 1 mark. So use different concentration of glucose. Fine. But in the experiment, we know based on what is given is 5% and 10%. You know what is the solution. Inform to the examiner what is the solution. So make your answer better. Use different concentration of glucose, which are 5% and 10% boiled glucose solution. The list didn't mention is boiled, but in the experiment, we need to use the word boiled glucose solution because why boil? After this, we'll explain. So use different concentration of glucose. Different concentration of glucose is manipulated variable. Different concentration of glucose is manipulated variable. I'm using what's given here, concentration of glucose solution. So use different concentration of glucose. Okay, let's make this better. Add the word solution. See how when I see something in the question, I add it in the answer. That's the thing. Get everything from the question, write back what's in the question. That's your marks. So use different concentration of glucose solution, which are 5% and 10% boiled glucose solution. We mentioned what is manipulated variable, and we also mentioned what is the manipulated variable that we use in the experiment. This is manipulated variable. Before I get the responding variable, I want to explain to the examiner what are the constant variables. So the constant variables, some of it we got from the diagram. We can see the 2 gram of yeast, you can see 15 ml. Another one that is constant is from here. The number 7 minutes. The duration of experiment is 7 minutes, also constant. So you can identify 3 constant in this experiment. You write any one of them should be enough. If you want to write more than one, also okay, but generally 1 for the constant variable. So I'll show you all 3. So the first one I'm mentioning here is fix the mass of yeast at 2 grams. You must use the word fix. Fix the mass of yeast at 2 grams. Must mention. Without the word fix, you don't get the 1 mark. So we don't have to explain why we are fixing it. We just inform the examiner we are fixing the mass of yeast at 2 grams. Or the others, what you can write, fix the duration of experiment at 7 minutes. Or fix the volume of glucose. Fix the volume of glucose solution at 15 ml. You decide. If you want to write all 3, also can. But generally 1 is enough. But if you feel you want to write all 3, right, go ahead. So fix the mass of yeast at 2 grams, fix the duration of experiment 7 minutes, and fix the volume of glucose solution at 15 milliliter. All of that I didn't create. I got it from the question. Just make sure you use the proper terms. If you're not using the proper terms, you may not get the mark. So I'm done with manipulative variable. I'm done with constant variable. The responding variable that we got from the table is count and record number of air bubbles released in 7 minutes. I repeat, if it's responding variable, you must mention the device. Counting the number of bubbles, we are using eyes to count and maybe the brain to count. So we can't put that as a device. But anything else, measuring the time, mention the stopwatch, measuring the distance, mention the ruler meter, measuring the temperature, mention the thermometer. The device must be mentioned. No device, consider answer not complete. You won't get the 1 mark. So 1 mark for manipulated variable, 1 mark for responding constant variable, 1 mark for responding variable. The question also mentions explain about the precautionary measures. Most of the time, the experiment will have many precautionary measures. This is unguided experiment. Unguided experiment, you just have to state what is the precautionary measure. If it's guided experiment, they can actually ask you what is the precautionary measure and explain why you are doing this precautionary measure. You might need to explain why you are doing it. So what I've done here, because we are treating this like a revision, I don't have a separate video for unguided. You understand this video is more than enough. So I'll combine. So any part where I need to explain a bit more, I'll explain a bit more. Like here, I'm only supposed to give you what is the constant variable, but I'm also going to explain to you why you need to do, not constant variable. What is the precautionary measure? Not only I mention what is it, I'll also explain to you why you must do this precautionary measure. The first precautionary measure that we are going to explain is make sure the keyword is not keyword, but the term that you must have. Make sure. Make sure the mixture of yeast and glucose solution is stirred gently. When you first mix the glucose and the yeast, to make sure they mix evenly, you're supposed to stir it with a glass rod. That's why they've given you a glass rod in the list here. You can see the glass rod. So glass rod is given basically for you to mix the yeast and the glucose solution. But you have to stir it gently and don't stir it too long, because when you stir the mixture, oxygen from the atmosphere will dissolve into the mixture. So we don't want oxygen from the atmosphere to dissolve into the mixture. So stir gently and for a while and then immediately continue with the experiment. Prevent atmospheric oxygen from entering into the mixture. So at the moment, to prevent, so I repeat, what I've given here, the second sentence is the explanation. This is what you need to write for unguided experiment. You don't have to explain why you are doing it. Actually, again, if you want, you can always write it, no harm. So make sure the mixture of yeast and glucose is stirred gently and don't take too long to stir. After a while, you continue with the experiment. Why? To prevent atmospheric, atmospheric oxygen from dissolving into the glucose solution. So this is the reason why you do it. This is what you do during the experiment. But the precautionary measure, not only this, there is second one, which is make sure the mixture of yeast and glucose solution is covered with something called as paraffin layer. Again, you don't have to create this because it's already in the list. They already mentioned to you is paraffin. Beaker that contains paraffin oil. So that's the beauty of paper 3. From the list of material and apparatus, you can get almost everything that you need. So why do you cover it with paraffin layer? The same reason why you didn't stir the mixture quickly. Because atmosphere contains oxygen. If you don't cover the mixture with paraffin oil, which is a barrier, they prevent oxygen from dissolving into the mixture. So in here, to prevent atmospheric oxygen from dissolving into glucose solution. Again, this is what you do, this is why you do. Both of these is related to oxygen, atmospheric oxygen. One more is related to also oxygen. Make sure the glucose solution is boiled and cooled. There is two reasons why you boil the glucose solution. One is the glucose solution may contain certain microorganisms that might affect the experiment. So by boiling the glucose solution, we can kill the microorganism. We can sterilize the glucose. But more importantly, we have to make sure the glucose solution we are using doesn't contain oxygen. So to enable the oxygen to be released out of the glucose solution, we have to boil the glucose and then you have to cool it, because if you're using a hot glucose solution, you put the yeast will die. So we boil it, we cool it, and then we mix it with the glucose solution to make sure the experiment is efficient. Maintaining a room temperature should be good for the mixture. So what we do in here, the last explanation, why do you boil it? To sterilize the glucose and also to release any glucose, any oxygen in glucose, so that the yeast can do fermentation. Yeast only do fermentation if there is absence or there is no oxygen. Low amount of oxygen or no oxygen, then only yeast will do fermentation. So we make sure oxygen is not present. Cover the mixture with paraffin, use boiled glucose, and when you stir gently. I've given you 3 precautionary measures. You decide which one you want to use. Any one of them should give you 1 mark. By right, for guided, you don't have to explain. Sorry, for unguided, you don't have to explain why you're doing it. If you want to write, go ahead. So better safe than sorry. If you feel you want to write it. So this is for how to conduct the experiment. If it's guided, the whole procedure will be given in the question. As I already mentioned earlier, in my website, I have both questions. I'm discussing unguided. There is also another guided paper. Download that and you can go through how the experiment is conducted. In here, the first 4 marks is basically to make sure the students know what is the manipulated variable, what is the responding variable, and what is the constant variable. Why? And what is the precautionary measure? Why you must know all the variable? That is what you're going to use for every answer after this. So first question A is how to conduct the experiment. Use the right terms, and make sure you identify the variables. This is considered easy for marks. And after that, we go to question B. Let's read question B. Carry out the experiment by using 5% glucose solution. State the observation on the number of air bubbles released in 7 minutes by using stopwatch. Actually, you can also get the responding variable from here. Earlier, I said you can get it from the table. Looks like you can also get it from the next question. It's all over the place. You know where to find it, you can get your marks. So you're supposed to carry out the experiment. You're supposed to state the observation. So by right, this question should come after you already complete the table. So this was the table that we showed you earlier. So this is the table. So if you conduct the experiment, let's assume now, just took these numbers, most probably this is how the number should be arranged, should be between like 20 to 25 or so. I'm just randomly pick number 8. You don't randomly pick. You conduct the experiment and get the answer. I didn't conduct the experiment, so I'm assuming it's going to be something like this. So when you write the observation, there is only, like in here, there's only 2 experiments. So you have 2 observations, choose any one. If let's say you have more than 2 observations, like here we have only A and B. Let's say now there is another test tube C, for example. We don't know how the question is going to be. We don't know whether this question coming out or not. But let's assume if this one comes out, and instead of giving 2, they give you 3 concentration, 5%, 10%, 15%, for example. So if 3, when you have 3 data, you must choose the highest and the lowest responding variable. So if I write it at the side here, if it's more than 2, you must use the highest or the lowest. So choose between highest or lowest RV. For this experiment, you have only 2. So you can choose any one, doesn't matter. But if it's more than 2 observations, you have more than 2, maybe got the C, then you choose the highest and the lowest, not manipulated, highest and the lowest responding variable. For example, let's say the next one is 35, for example. Then what you must do is you must choose either this one or this one as your observation. Choose the highest or the lowest responding variable. For this experiment, never mind, there's only 2, you can choose either one. So that's what I have done. That's what I have done. Let me just cover all of these answers. Oh, you already see before I, never mind. Okay, so let's see. So this is the results that I'm, observation that I'm giving. In test tube A, which contains 5% boiled glucose solution, number of air bubbles released in 7 minutes is 20 bubbles. I've already color coded for you. What's in green is manipulated variable. What's in red is the responding variable. So the green one is manipulated variable. The red one is responding variable. This is what I mentioned many times. Just make sure you have all the variables in the answer. That's all. And what is 20 bubbles? 20 bubbles is supposed to be the data. Make sure you have manipulated variable, responding variable, and also the data. That should be enough to get you the marks, 1 mark. So in test tube A, which contain 5% boiled glucose solution, number of air bubbles released in 7 minutes is 20 bubbles. Because it's observation, you don't explain anything. Just write what you see. What you see in the table, write back. That is the answer for the exam. So after you write the observation, the next question, questions B2 is explain your observation. Now they're asking you to explain. Guided paper, they'll ask you for the inference. Inference basically means explain your observation. So you told the examiner, if you use 5% boiled glucose solution, you get 20 bubbles. Why you get 20 bubbles? That is, the examiner wants to know, explain. So what you do is, you just explain your observation. Yeast in test tube does alcohol fermentation in no oxygen condition. I've added the word alcohol. the question just used the word fermentation. Now, where did I get the word no oxygen condition? It's from the question. If you go right at the top here, they already mentioned either the condition is limited or no oxygen. So I just use back what's in the question. What they do, they are doing fermentation. I added the word alcohol because they produce ethanol. We know that from theory. If you didn't have alcohol also, never mind. Yeast in the test tube does alcohol fermentation in no oxygen condition. This explains what the yeast is doing, but it doesn't explain where these bubbles came from. They are doing alcohol fermentation, fine, but where did the bubble come from? You must explain where the bubbles are coming from. That's why we have another sentence. Fermentation releases carbon dioxide, which are released as air bubbles. So you told the examiner, if you give glucose, the yeast is going to release bubbles. That's what we are explaining. By using boiled glucose, the word here is boiled. By using boiled, we are informing the examiner there is no oxygen. Glucose will be used by the yeast to do fermentation. What is produced is ethanol and also carbon dioxide. Carbon dioxide is released as air bubbles. Nothing hard in this experiment. Always we give one observation and we explain that one observation. That's the next question. This is supposed to be inference. Explain the observation is basically inference. What is the next question? The next question I think is fill in the table. Conduct the experiment, repeat the experiment using 10% glucose and complete the table. So this is how the table looks like. As I mentioned, I didn't conduct the experiment. Most probably these are the numbers that you get. 18 bubbles and 29 bubbles. So once you already fill in the table, take note of the decimal point because this is bubble. Because it's bubble, I don't have a decimal point. If you're measuring, let's say, distance. So if you're writing in centimeter, you're writing in centimeter, but the ruler has millimeter, then you must write 12.1 or 12.2. Decimal points are important. If your decimal points are not correct, then another 1 mark will be lost. When you complete this table, the decimal point, make sure it follows what you have in the experiment and make sure all the decimal point is same. For example, let's say there is some other experiment. This is 18.5. Let's assume it's 18.5. And here you wrote 29. Consider you're not following the same decimal point. That 1 mark you don't get. If here you use 1 decimal point, here also must use 1 decimal point. Even if it's 0, make sure it follows the previous decimal point. They're very particular when it comes to decimal point. And also take note that you can get the unit from the table heading. So the percentage is the unit for concentration of glucose. In here, no unit given, but I'm going to use bubbles. After we complete the table, the next question is calculate the rate of yeast reaction. Most of the time, this will be in the table. That means there'll be another column on this side that will have the heading, a rate of reaction. I have separated the question. I want you to show me the working. So basically what you do is, and now the formula is not given. Sometimes what they do is they don't give the formula, but they give you clue. The clue given in the question is calculate the rate of yeast reaction for boiling tube A and B in number of bubbles released per unit of time. So that tells us the rate of reaction is number of bubbles released divided with the time taken. The time taken is 7 minutes. So always if you are doing calculation, show the working. I make sure I show the working. So the rate of yeast reaction in test tube A. This is the formula. So responding variable divided with constant variable. The value we got is 18 bubbles divided with 7 minutes. I always make sure I write the unit. Even though the textbook didn't mention bubbles, I'm writing 18 bubbles divided with 7 minutes. The answer we get is 2.6 bubbles per minute. Write the unit. Without the unit, you don't get any marks. This is for test tube A. For test tube A, I show the formula. For test tube B, I go straight into the working because I already write the formula earlier. So to save time, I just write the working and I get 4.1 bubbles. Realize that this is 1 decimal point. This is 1 decimal point. Follow the decimal point. Some students ask me, so how many decimal points I need? Maximum you go is 2. You need to use all of this data to draw the graph after this. The higher your decimal point, the harder for you to draw the graph. So the best is 1 or 2 decimal point. I can make this into 2 decimal point, but I didn't because I realized it will be hard for me to draw the graph. So I stop it at 1 decimal point. If I make it 2 decimal point, then I need to have a very precise graph. This question has a graph. So we stop at 1 decimal point. Maximum is 2. So all of these are considered easy questions. We are done with all of this. We can go straight to the next one. So the next question that we have right on top here. The next question that we need to explain is, based on the data in C, explain the difference in the rate of yeast reaction between the two glucose concentration. So the last question that we did, we already calculate that the rate of reaction in test tube B is higher. The rate of reaction in test tube A is lower. So the question wants to know why. Obviously, the difference between these two is the manipulated variable, which is the concentration of glucose solution. Because the glucose concentration is higher, the rate of reaction is higher. But we need to explain it in properly. So the first thing we are going to do, we are going to inform the examiner. The yeast reaction in boiling tube B releases more carbon dioxide compared to boiling tube A. The word more is important. This is an important keyword. And releasing more carbon dioxide is a responding variable. Releasing carbon dioxide is responding variable. So we are informing. Here got more responding variable, here got less responding variable. But responding variable got 2. The amount of bubbles released is responding variable. The rate of reaction is also responding variable. So I'm writing another sentence which means exactly the same, which is the rate of yeast reaction in boiling tube B is higher than boiling tube A. Both is responding variable. In this experiment, there is 2 responding variable. Rate of yeast reaction, the number of bubbles released. I'm informing the examiner for test tube B, the number of bubbles are more, the rate of reaction is more. Why more? That is our explanation down here. Boiling tube B contains higher concentration of glucose compared to boiling tube A. We are informing the difference is because of manipulated variable. A has less manipulated variable, B has more manipulated variable. So all of this is more. More carbon dioxide is released. Higher rate of reaction because there is more glucose concentration or higher glucose concentration. Your result shows B is higher than A, rate of reaction. Here we are explaining why. More CO2 release, higher rate of yeast reaction because higher concentration of glucose. After that, consider the main part of the experiment done. We have identified all the variables. We already conduct the experiment. We already explained the results. The difference in the results is because of manipulated variable. This one has less manipulated. This one has more manipulated variable. So this is the ending of the question. So what else they can ask you? The next question we are going to discuss is hypothesis. Now, the hypothesis is always asked in the guided question. This is unguided, but just in case I've given it here. So the question is asking for the hypothesis. Hypothesis must have manipulated variable and responding variable. What is the relationship between manipulated and responding variable? Most of the time, the answer will be just the combination of this. The higher or the lower the MV, the higher or the lower the RV. You must start the answer with manipulated variable. You must finish the answer with responding variable. Constant variable is not needed. Most of the time, you can use higher, lower combination. There are times where we cannot use higher, lower combination. For example, if we have only 2 data. Actually, this experiment also we have only two data, but most of the time, if it's two data, we cannot use this higher lower combination. We will use the compare. We use the word compare. For example, there is an experiment energy value of a certain type of food substance. Maybe we use cashew nut and groundnut, for example. Now, when you do the experiment, energy value for cashew nut, you get certain number, and for the groundnut, you get certain number. But you can't be, writing the hypothesis using this technique here. The higher the nut, the higher the energy value. Sounds so, sounds very funny. But in situations like that, where you cannot use the higher lower combination, that's when we use the word compare. Cashew nut has higher energy value compared to groundnut. So keep that compare in mind. Anytime you can't create a hypothesis using this technique, try to create using the word compare. For this one, we can use higher lower. So we just, the higher the MV, the higher the RV. That's all. So what we are going to write in here, the answer for this question. What is the hypothesis for this experiment? The hypothesis for this experiment is the higher the concentration of glucose, the higher the rate of yeast reaction. Realize that what's in blue is the manipulated variable. What's in green is the responding variable. So manipulated variable here I've given in blue color. The responding variable I've given it in the green color. So the concentration of glucose is manipulated. The rate of yeast reaction is the responding variable. What is the relation between these two? The higher the MV, the higher the RV. But if you realize in this experiment, there is two RV. One RV is the number of bubbles that are released. Another RV is the rate of reaction. So rate of reaction we already used. If you want, there is another answer. Exam time, write one enough. I'm showing you both. The other answer you can use is this. The higher the concentration of glucose, the MV remains the same. The greater the number of air bubbles released within 7 minutes, I'm using the experiment responding variable. What you measured in the experiment, the number of bubbles you counted. You can use that as the answer. I've given you two answers. Choose one. Not all the time you can use higher, lower combination. Sometimes you have to use the word compare, check the experiment, decide accordingly what you need to write. So, but the thing is, it must have MV, it must have RV. If you realize every single answer has MV and RV. Every single answer has MV and RV. Even here also, the table, we have MV and RV. Here also, we write MV and RV. That's the trick. Every answer, make sure you have MV and RV as much as possible. Add that two, you can get your full marks. So from there, the next experiment that you're supposed to, the next question you're supposed to do almost at the ending is by using the data in table 1, draw an appropriate graph to show the relationship between the rate of reaction in yeast against the concentration of glucose solution. Use the graph paper provided. I've already drawn the graph. The 3 marks for the graph is just take 1 mark for the x-axis, 1 mark for the y-axis, 1 mark for your graph. Many students for this question, they spend a long time drawing the graph. But they don't realize that the graph itself is only 1 mark. There's another 2 marks in the x and y axis. So complete your x and y axis. Make sure you use your right scale. Make sure you give the heading. Make sure you give the unit. Get the 2 marks because 1 mark for X, 1 mark for Y, 1 mark for the graph. Now, before you can even draw the graph, complete the 2 marks first. Many students don't do that. They are so focused on drawing the graph, they don't do the X and Y properly, end up they lose 2 marks. So the first thing you need to do is choose the right scale. General rule is the graph must be more than half of the graph paper. So draw as large as possible. General rule, it must be more than half of the size of the graph paper. So I try to maximize the x and y axis. So first we go straight to the y axis. So the y axis is at the bottom here. So I know the value is only 5% and 10%. So I make the graph, the y-axis as large as possible. So it's almost using the whole of the graph paper. Don't forget to label the origin 0, 5, 10. Make sure the scale is same. So I'm using 4 grids or 8 grids. Here got another 8 grid. So the scale must be correct. The label must be correct. Here also, concentration of glucose solution, percent. The unit also must be given. So I secure my X. I make sure my X axis already correct. Then I go to the Y axis. Now this is how the Y axis should look like. So my value that I need is, I need to go until 4.1. The value we got earlier is, highest is 4.1, lowest is 2.6. So I need to have from 0 to 5 minimum. So that's what I did here. So I choose every 4 grid as 1, and I go all the way up to 5. I draw the graph as large as possible, and I make sure I write the heading, rate of yeast reaction in bubbles per minute. What many students do is they write here 1, 2, 3, 4, 5. If you do that, you lose 1 mark because you must follow your calculation. Your calculation is 2 decimal point. Your graph must be 2 decimal point. A simple mistake that students don't follow, they lose 1 mark for the x and y axis. Decimal point must follow the calculation that you gave earlier. So I bring it as large as possible. Now to plot the graph, the two values that I have is 5% is 2.6, 10% is 4.1. So we first draw the 5% one. So we go to the 5%. This is the 5%. And what I do, so I know the line is here. This is the 5% line. So we go to 2.6. The value 2.6 is somewhere here. And this is the intercept. I already labeled. You don't have to draw this. I'm just showing you how to get this intercept. That is for 5%. Now for the 10%, I should get 4.1. So 4% is 4.1. Again, I just draw a line to make sure I get the correct one. And then I just remove this, is not needed. When you have 2 points only, no, it's a straight line. If you have 2 points, it's a straight line. If you have more than 2, then you have to make sure you draw either a straight line, or if it's a curve, make sure it's a smooth curve. Now, one mistake that many students will do is, they will, for this graph, what they'll do is, they'll have another line here. They will have another line here. From here, they bring to 0. Many students do this. Now, you might have to do that for physics. You might have to do that for physics, which is you extend the graph. You don't do that for bio. For physics, normally, again, you have to check with your physics teacher what you're supposed to do. But for bio, you cannot extend the line because we don't know how the line is going to be. Extend the line, 1 mark gone. This graph, might go like this, might go like this, might go like this. We don't know. If you don't know, don't draw. So just draw what you know. You know 5% is 2.6, 4% is 4.9, sorry, 10% is 4.1. Draw that enough. Don't simply extend the line. For other subject, chemistry and physics, check with your physics teacher, check with your chemistry teacher. For bio, you extend 1 mark gone. We draw the largest possible graph. Don't extend the line, meaning the line must stop at the point and start at another point. If it's 2, straight line. If it's more than 2, if you draw a curve, make sure it's a smooth curve. Check the graph. Most probably you'll get a simple graph. Actually, this question didn't tell us what graph to draw. We are drawing a line graph. If you want to draw a histogram graph, also for this can, because it's only 2 data. But the thing is, most of the students are preoccupied in drawing the graph. They lose sight of the x and y axis. Secure your X and Y, label everything, get the 2 marks, then start to draw the graph to get the remaining 1 mark. At the same time, don't waste too much time because you have to answer other questions also. So don't waste too much time on the graph. Get the X, get the Y, get the graph, move on to the next question. After you get the graph, so this is the graph. So the number down here is 0, 5, and 10. Another thing that students tend to miss is to label the origin. Many students miss that. So we are done with the graph. This is the graph we already draw. After we draw the graph, we go back to the last question. So there is another question here. And this question is, okay. This last question is always prediction. Prediction, basically what they are going to do is, every answer before this, generally we focus on the manipulated variable and responding variable. Rarely we use the constant variable because it's constant, it doesn't affect the experiment. So the last question is where the constant variable will come to light. So the last question is where we normally change the constant variable and then ask you to predict. So a student repeated the same experiment as boiling tube B, but does not add paraffin oil to the boiling tube. So in here, what they're doing, they're changing the precautionary measure. We can also change the, for example, I can simply ask you a question. The constant value is 2 grams of yeast. So I can ask you, if you use 3 grams of yeast, what happens? The other constant is 15 ml of glucose solution. What if I use 25 ml of glucose solution? So that's how the prediction question will be. Change the constant variable, predict what's going to happen. The problem with the students is they will predict, they will give the right prediction, but they always fail to provide the data from the experiment. The question mentions you are not using paraffin oil. Earlier, we already told paraffin oil is to prevent atmospheric oxygen from dissolving into the glucose solution. If you didn't use paraffin oil, the glucose enter into the glucose solution, diffuse into, dissolve into the glucose solution. Now the yeast can do aerobic respiration. Aerobic respiration, glucose breakdown is complete. They produce more carbon dioxide, meaning more bubbles will be released. That's what the students like. When the yeast do aerobic respiration, more air bubbles are released. The problem with that answer is there is no data. Always use data. Where you got the data? You got the data from here. Earlier, you told the examiner the number of air bubbles released in B is 29. Don't inform the examiner there is more bubble. Tell him it's more than 29. Use the data from the experiment when you make the prediction. Use the data. Use the data in the answer, then you can get your full mark. So for this question, instead of telling is more bubble, use the data. So the answer I'm going to give here, the number of air bubbles released in 7 minutes is higher than 29 bubbles. Where did I get 29 bubbles? From the experiment. Data from the experiment. Okay, fine. If you have 29 bubbles, why? For prediction, you must explain whatever prediction you make. So the prediction I'm making here is higher. Why? As I explained, oxygen will dissolve into the glucose solution. Aerobic respiration will happen. So without paraffin oil layer, atmospheric oxygen gas dissolve into the glucose solution. Yeast does aerobic respiration by using oxygen. More carbon dioxide gas is released. More keywords in the answer. This is, we didn't explain earlier. I told you when I was explaining how to control the variable, I told you when it's about constant variable, we mention what is it. We don't explain why. We explain when they ask. So like here, they are asking. So this is actually the opposite of the function. Paraffin oil supposed to prevent gases dissolve. Now don't have, so the gases dissolve, specifically oxygen dissolve. That's the end of the question. But I'm adding in one more question here. In unguided, this is unguided. It ends with this prediction. If it's guided, the last question is normally based on this experiment, deduce what is something. They ask you to deduce or state operational definition of something. And many students are normally confused how to write this operational definition. They normally write what they learn in class. Now, practical different, what you learn in theory a bit different, the way you answer it. This question, operational definition, the question will be like this. I've added one question here. Not this. Oh, yeah. This is just to inform you. You can scan this to get to watch this video that you're already watching now. But if any of your friends wants to watch the video, just forward them the question paper. They scan this, they can watch the video. I have the BM version and also English version, free of charge. No need to pay anything. Okay, community service for the students. Okay, so here, let's say the question is based on this experiment, state the operational definition of fermentation. State the operational definition of fermentation. Let's assume the question was like this. State the operational definition of fermentation. Many students are very confused when they see the word operational definition. And many of them, like for example, the question asks you operational definition of photosynthesis. What they'll do is they'll write back the definition of photosynthesis. Process that produces complex organic molecules from simple organic molecules by using sunlight energy absorbed by chlorophyll. That is the definition of photosynthesis they learn in class and they use it for this answer, not accepted. They didn't ask you what you learn in class. They are asking you in this experiment that you conducted, what is photosynthesis or what is fermentation? The answer must be from the experiment, and the answer is super easy if you know the technique. The answer is, the first answer is this. Question is responding variable plus constant variable. Question means this one, what is fermentation? So fermentation is responding variable plus constant variable. The second sentence is the question, which is fermentation, is affected by manipulative variable. Perfect question. For me, it's a perfect question because we start with the 3 variable, we end with the 3 variable. We start with the constant variable constant, we finish with the constant variable not constant. Perfect beginning and perfect ending, my view. So just use this technique, answer the question. So the first answer I'm going to write will be this. Fermentation is the release of air bubbles from 2 grams of yeast in 7 minutes. Fermentation is the question. Release of air bubbles is the responding variable, green color. Constant variable is 2 grams and 7 minutes. I have 2 constant variable, 2 grams and 7 minutes. Fermentation is the release of air bubbles from 2 grams of yeast in 7 minutes. The second sentence is the question is affected by manipulative variable. So fermentation is affected by concentration of glucose. Use this technique, automatically operational definition becomes easy. I don't know about other subjects. For bio, you can just use this if you get a guided question. If it's unguided question, this question normally don't have, they ask you this question. If you realize both this question and this question, what we are doing, we are changing the constant variable. That's the only one didn't change throughout the experiment. The last one, we changed constant also. The only thing constant in the world is change. I'm blabbering, never mind. Okay, we are done. So we are not sure what is the question for the exam. So if this one comes out, consider very good. We already discussed it. Now, if let's say in the actual exam, some other experiments comes out. Don't worry. Go to the list of material and apparatus. You can see the list in the question. You can see them in front of you. You can create the experiment on its own. If needed, what you can do is, I think you have the Perker book, the lab experiment book. Just go through once. Just go through once how the experiments is conducted. Just go through once. Just to remember general idea. What is how to conduct the experiment. You see the list, from the list of methyl apparatus, create the experiment. Not hard. A lot of information you can get from the question itself. So I consider the only problem I see in the experiment is something might go wrong when you're conducting the experiment. So when you are conducting the experiment, something might go wrong. That one we don't know, a bit unpredictable. But touch wood, let's assume everything goes on normally. So once the experiment is done properly, the rest of these questions that we discuss, you must get full marks. Don't come back and give excuse. I don't know how to answer this. I don't know how to answer that. Paper 3 is a bonus question. Follow the instruction, get the results, follow the techniques I mentioned and what your school teacher already explained, get the full marks, no excuse. I'll meet you when I'm discussing paper 2. Paper 3 done, get your full marks. See you again.